we'll start. So I'm going to talk about membranes and membrane proteins today. And this actually builds on one of the earlier lectures that Eric has given and builds on what students have learned about amino acid properties, secondary and tertiary structure, and structure-function relationships in globular proteins. And so we're going to learn what is basically different between globular protein and membrane proteins. And so to go through this, we need to start with the membrane itself, because to understand membrane protein function, we need to understand its environment. And so the first part of the lecture will be about function and structure of the membrane, and then we'll move on to understanding what are the structures of membrane proteins, and then we'll talk about function and how structure supports function for two specific classes of phenomena that are important at the membrane: transport and signal transduction. Okay, so like I said, we start with the membrane. And I think the way this is introduced in the book I was using is really cool. Actually, I've never heard the story that way. So the idea is that before the end of the 50s, the only, let's say, imaging technique that we had was light microscopy. And as you might know, the membrane is a very small dimensional object. So at that point, we couldn't even image it or see it. But towards the end of the 50s, electron microscopy became available. And so this is an image of a membrane of a tissue that was stained with osmium, heavy metal. And what is important to see is this laminar structure here at this level. So the point is that osmium comes and partitions preferentially in places in the membrane, and then what you can observe is this kind of structure. So this would be what separates the inside of the cell from the outside of the cell, and so then it's the plasma membrane. But interestingly, when you observe different types of tissues, like in this type of tissue, you can see that not only do you see the plasma membrane that delineates, again, the outside of the cell from the inside of the cell, but you also see intracellular membranes. And this was, I guess at that point, surprising, because it means that there are not only the same kind of structure of membranes not only separates the cell from the outside, but also inside compartments that presumably fill out some physiological function. So this is called here the sarcoplasmic reticulum. And the thing to notice is that this periodic structure that contains two dark leaflets and a less dark leaflet in the middle is the same in both cases. So like I already said, the main role of the membrane is compartmentalization. And that means the main role of the membrane is compartmentalization, and that means that it isolates the inside of the cell from the outside of the cell, like I just said before. But that's not the only role that the membrane fulfills. There's actually biochemical activity and other types of phenomena that are going on at the level of the membrane. And in this lecture, like I said, we will focus mainly on transport and response to external signals, but it's worth knowing that membranes also play a role. For example, they allow biochemical activity to happen inside of the cell. They can also allow cells to communicate with one another directly. That's when you need really fast transport properties. So it's the case in heart cells, for example, where then you get direct connection from one cell to the other, and this is made by the specific shape of the membrane. And we can have also energy transduction. So I mentioned just earlier that you might have intracellular organelles, and in the case of a plant cell, which this is depicting, you can see that you have different types of organelles. This is a mitochondrion, and then this phenomenon happening creates energy for the cell, basically. This is a chloroplast, and so then this biochemical activity is happening inside of the chloroplast in so-called thylakoid membranes that are inside of the chloroplast. So like I said, we're going to be focusing on how compartmentalization happens, and then also on transporting solutes. Here you can see as depicted, water can come in and out of the cell. and ions can also be transported, and we'll cover the physical basis for this. And then, as I said, towards the end of the lecture, we'll mention signal transduction. This is an example of signal being transduced, where you have an extracellular compound that binds to the to a protein, and this causes a cascade of events that is important for the cell. We'll cover the basis for this. Okay, so like I said, and when I asked the students in the class last week, I asked, do you guys know already why this periodic structure happens? I think in this audience, you know a bit more, but now these are the first experiments that were showing this. So membranes are made actually of lipid, essentially, and the lipid is a molecule that contains mainly two parts. Hydrophobic part that is made of long hydrocarbon chains, and a hydrophilic part that is made of a smaller polar or charged head group up here. And because of their biophysical properties, then membranes tend to assemble in a way that is favorable thermodynamically as other type of assemblies. And this means that the polar head will have a tendency to face polar media, like solvents, while hydrophobic tails will have to isolate themselves from the solution to avoid unfavorable contacts. And this quite cool experiment by Groja and Grendel was one of the first to show, while we had no idea of the physical basis of structure of the cell, of the membrane, that membranes must be made of a lipid bilayer. So the way this apparatus works is you break up a cell. In this case, they were using red blood cells, and this is because mature red blood cells, they don't contain any intracellular organelles. So basically, the only membrane that you have in this kind of cell is the plasma membrane. And so if you break up the membrane, and then you put it on this kind of apparatus that contains water in the bottom and a movable barrier here, to obtain one single layer of lipids, you can count that this surface covers about twice the one that you measure in a red blood cell. So this means that then the bilayer must, or the membrane must be made of two layers. And there were kind of many errors made in this experiment, but all the errors kind of canceled each other out, and it turned out that it was correct that most of the membrane was actually made of a lipid bilayer. Soon after, it was realized that the surface tension that you expect from a bilayer is quite different from the surface tension that you get from a cellular membrane. And the hypothesis became that there must be other compounds that are involved in making the bilayer. And like Eric said in the first lecture, if something is doing something in biology, it's a good idea to assume it's a protein. So I guess that was the rationale behind. Protein must be making up something in the bilayer and then changing the surface tension to make it the one of a plasma membrane. And that was the model that was in favor in the early part of the 20th century. And we know that it's wrong nowadays, but it's not actually that wrong. So the idea was that there must be protein molecules that are coating the membrane, basically. And this allows to fulfill one of the roles also of the membrane, which is to allow to transport solutes across the membrane. So see how there you would have kind of a protein molecule that makes up a pore that then would allow solutes to go through. So this was wrong, but not that wrong, because we do know that pores are made by proteins a lot. And the model that we currently have, and that dates back to the 70s, is the fluid mosaic model. And so this is a depiction of it. Like we said, most of the membrane is made of a bilayer of lipids, as you can see here. One thing that is highlighted in this picture is that the lipids that make up the intracellular leaflet and the extracellular leaflet are different, and this is important to for cellular function. And then within the bilayer, you have proteins, but not proteins that kind of coat the surface, proteins that make up an assembly on their own, and they're inserted into the bilayer, as you can see in different ways here. And the crucial point here in this notion of fluid mosaic model, mosaic we've just covered, so it's this assembly of different species. But the key word is fluid. So fluid means that it's not a static assembly where you just have lipids and proteins in between. The lipids can diffuse within the membrane, and the proteins can also diffuse within the membrane. So I don't have a movie, but you should imagine that this is a dynamic thing, where also this means that lipids can come and alter the function of proteins, and proteins can come together and apart and create new functions, basically. Good. So to go back quickly to the structure of lipids, as I said just earlier, usually lipids are made of a polar or charged head group and hydrophobic tails, which is what you see on this picture. This picture is meant to highlight that there are many different types of lipids, different chemical compositions that make up lipids. So this simpler one up here has a negatively charged phosphate group with two long hydrocarbon tails, and the yellow part would be the linker region that allows to couple chemically the two parts of the lipid. And then chemically, different types of groups can be attached to this phosphate group, and this increases then the complexity and the lipid chain. This is a different family, sphingoside and sphingomyelins, but you can see that basically the structure remains the same. So a polar head and a apolar tail. And so this chemical complexity makes it possible for bilayers to then be quite varied. We talked already before in the fluid mosaic models that you would have different lipids in the extracellular leaflet and in the intracellular leaflet. But not only that, you can imagine if you have lipids that have big head groups and small tails, they will have a tendency to adopt a curvature where the curvature is towards the tails. And if you have lipids that have a small head and a big tail, then they would have the kind of opposite curvature. So with the type of chemical diversity, you can create not only different types of structures, but also act on membrane proteins in different ways, and we'll come back to that in a minute. Another type of compound that is really important in the membrane is cholesterol, which is depicted on this figure. So you have a kind of regular phosphatidyl choline lipids in gray, and then in green here you have the cholesterols. So I don't have a very good picture of this, but cholesterol is quite different from the other lipids. It's the similar philosophy of having a polar head group and an apolar body, so it orients always with the polar head group facing towards the solution and the hydrophobic body facing towards the inside of the membrane. But cholesterol has a very rigid ring-like body, so it intercalates between lipids. You can see that the other lipids tend to curve around it and to accommodate the cholesterol. So one of the main effects of cholesterol is that it rigidifies bilayers because it has this rigid body and small head group. So this explains why the membrane can act as a delineator and separate the inside of the cell from the outside of the cell. You saw that it's the hydrophobic body that will prevent solutes from going from one side of the membrane to the other. And by the way, I should mention that you may have noticed now that you know what is the structure of the bilayer, why the first picture with the electron micrograph had this shape of dark leaflet, a light leaflet, and a dark leaflet, right? Osmium partitions more into the head groups, and so this is why you were having more density and a stronger signal at the head groups than inside of the body. So this explains that, and nature is beautiful. So now we know what the membrane looks like, and we're going to talk for a little bit about how this guides membrane protein structure. There are essentially 3 types of membrane structures. The ones that will span the entire membrane are called integral membrane proteins. They're usually made of alpha helices. So we've talked in previous lectures about how alpha helices form through the formation of hydrogen bonds between an amino acid in the sequence and the one that is a few amino acids later. Usually if it's an alpha helix, it would be the amino acid that is 4 positions downstream in the sequence. So this is one helix that is spanning the membrane, and you have integral membrane proteins that are made of several, a number of helices. These ones are called peripheral membrane proteins, and they're made of domains that are associated with the membrane through non-covalent interactions, and they interact either with the extracellular leaflet or with the intracellular leaflet. And then we have lipid-anchored proteins, where the protein chain is directly covalently linked to a lipid, and so it's associated then with the membrane through a covalent interaction. Especially in eukaryotic proteins, reality is much more complicated, and in general you not only have integral or transmembrane domains, but you also have peripheral domains, and so this on this figure you could see that you have an intracellular domain, an extracellular domain, and a transmembrane domain, and this is usually the case in more complex membrane proteins. We'll see that in a few examples in a minute. So remember from the fluid mosaic model what happens. You have these integral membrane proteins specifically that span the entire membrane. And one of the key aspects is that the interaction or the interface between the membrane protein and the lipids must be tight enough so that there is no leak going on at the level of the interface, basically. So this is a specific protein called aquaporin, which we'll talk about in a minute. And recent experiments show more and more that lipids are tightly associated with the membrane protein. Here you have the lipids chains that are in yellow, and those would be the lipids that are interacting and tightly bound directly to the surface of the membrane. And one of the key functions for lipids that is emerging and becoming more and more important, as we see in kind of current research, is that lipids don't only play a role as a scaffold for membrane proteins, they regulate membrane protein function. And this is a really hot topic right now, and one that we actually are particularly interested in. I'll talk about this a bit more going forward. Okay, so this is maybe the most important slide about membrane protein structure. I want you to pay attention to the type of amino acids that usually make up transmembrane regions. So this is the example of glycophorin A. It's an integral membrane protein, that is made of a single transmembrane helix and an extracellular domain and an intracellular domain. And here you have a zoom in onto the helical part. And so that would be the protein, the amino acid sequence that makes up this helix. And in red here are depicted amino acids that are hydrophobic in nature. In blue are depicted amino acids that are hydrophilic or polar. and in gray are depicted glycines. And here in green you have positively charged amino acids, lysines and arginines. What this picture shows is that there's clearly a difference in the makeup of the protein chain that is in the transmembrane region and that is in the extracellular domain. And this should not surprise you if you studied the chemical properties of different amino acids. Positively charged amino acids should be happy in a negatively charged environment, should interact favorably with a polar and negatively charged environment. And in fact, the lipids that make up the intracellular leaflet of the bilayer tend to be negatively charged. So then it makes sense that you have these positively charged residues here inside of the cell. And the hydrophobic residues tend to partition into the lipid bilayer core because they interact favorably with the hydrophobic chains of the lipids, not too surprisingly. So now the question is, does it make sense that you have also polar residues in a transmembrane region? What you can notice from this example is, first of all, that the polar amino acids tend to be not quite in the middle of the bilayer, but that they tend to be more towards the head groups. And this is because these regions are less unfavorable to hydrophilic residues. And this comes from the chemical property of this yellow linker I was showing a few slides before that is in fact more polar. But still here you can see that there is one threonine, so one polar residue that is in a transmembrane position. And so while this would be unfavorable to have a single threonine residue in the middle of the membrane, it's important to notice that it comes with the rest of the chain. So it is in fact possible to bury a hydrophilic residue in the bilayer, but you have to consider the balance of all the interactions. So if there are many hydrophobic residues that are buried in the membrane, then you can have also hydrophilic residues that are buried, as you can see here. And then the last part I want to highlight is the presence of these glycines. You've learned previously that glycines are small amino acids and they are very flexible, so they allow for alpha helices to break. And so why would it be favorable to have glycines in a single transmembrane helix is not clear. But if I tell you that glycophorin A is most of the time associated as a dimer, then you can understand why it would be favorable to have glycines, right? So instead of having two parallel helices, then you can have slight bends that are introduced into the bilayer, and then the two helices can interact together quite favorably. So this tells us that membrane proteins are usually hydrophobic, but you can have exceptions to this rule. And this brings us to what do we do when we want to study protein structure? So as has been said in earlier classes, to determine protein structure, you might want to do, whether it's X-ray crystallography, cryo-EM, or even NMR, you need to isolate and purify your protein in quite large quantities to be able to get enough signal. So what would happen now if I take my glycophorin A protein from before and I just strip it away from the hydrophobic environment and I just put it in solution? Because it's made of all this hydrophobic amino acids, then it would unfold, right? And it would kind of form an unstructured assembly where all the hydrophobic side chains tend to assemble together in the middle and expose only the hydrophilic ones. And so determining the structure of this would not be so informative because it would be so far from the structure through which it's functional. So the key here to determine membrane protein structure is to solubilize the protein to be able to concentrate it in high enough quantities but still retaining its native structure. And this is done typically with detergents. And if you look at the chemical structure of these two detergents that are depicted here, basically you'll recognize that they're at this level quite similar to lipids in the sense that they have a hydrophilic head and a hydrophobic tail. So by then solubilizing a membrane protein into a detergent, you can arrange the detergents into these so-called micelles, where the hydrophobic side chains of the protein will be in contact with the hydrophobic tails of the lipids and of the detergents, and the head group would be facing outwards. And in that way, you've preserved, hopefully, more or less, the native structure of your membrane protein, and you can do structure determination as you would do with a globular protein. Based on what I just said, it's interesting to notice also that we can quite easily predict transmembrane regions from sequence. So we know that there will be mostly hydrophobic amino acids that will be in transmembrane position. And so you can draw these so-called hydropathy plots where you calculate the free energy of transferring an amino acid from a nonpolar solvent to the aqueous medium, and this would be positive for hydrophobic residues, and this would be negative for hydrophilic residues. And this is the hydropathy plot that you would get for glycophorin, the protein that I just talked quite extensively about. And you can see that there's only one region here between amino acids, roughly 70 and 90, that is predicted to be a transmembrane region, and that is based only on its free energy of transfer from a hydrophobic medium to a hydrophilic medium. This works really quite well. You can predict transmembrane sequences from sequences quite reliably there are a bunch of servers that are available to do this. It's a quite reliable predictive method. I mentioned earlier, if you remember the glycophorin plot, that the positively charged amino acids were located inside of the membrane, and that is because the negatively charged lipids have a tendency to be located inside of the membrane. And so there's this so-called positive inside rule that was coined by our colleague Gunnar van Heijnen from Stockholm University, that tells you that you can recognize the intracellular domains from whether they have more or less positive amino acids. So in the case of glycophorin, which was a single pass transmembrane protein, the positive inside rules tells us that the N-terminus is intracellular and the C-term is extracellular, I believe. And then finally, the last thing I want to say about this protein structure is that we discussed quite extensively how you can have polar residues that are in transmembrane position based on the overall balance of the interactions. But it's even possible to have charged residue in the transmembrane region, and this is usually due to packing between helices. So if you have several helices that pack together, some of them bringing negative charge and some of them bringing positive charges, then you can isolate the hydrophobic surface to the outside of the structure and have all these polar and charged amino acid inside of the helical packing. And we'll come to an example of this in a minute, that's why I mention it right now. A quick note on other types of integral protein structures that are usually kind of forgotten about because they're less abundant. And these are so-called beta barrels. So the proteins that I talked about up until now were made of either one helix or several helices. This is a different type of assembly where the protein chains arrange in long beta strands that arrange into beta sheets and form these so-called barrels. So basically, if you were looking at it from the top you would see an empty kind of barrel where the side chains of the amino acids are oriented to the outside of the barrel, and typically most of these amino acids are then also hydrophobic in nature. So far, these type of proteins are only found in the membranes of bacteria, mitochondria, and chloroplasts, and not in the plasma membrane. And I won't say much more about this type of assembly. It goes much faster when you don't ask questions to people. Okay, so based on what we know now about membrane structure and membrane protein structure, we're going to cover two types of phenomena happening at the membrane, like I said, membrane transport and signal transduction. So, like we said, most solutes are polar or charged, and so they are basically not able to cross the membrane easily on their own. There are that way different types of transport. We can basically divide transport across membranes into 4 different types. First, there's the distinction between passive transport and active transport, and this the difference between these is whether the solutes are being transported down their electrochemical gradient, which I'll come back to in a minute. So you can see here that species A is more abundant outside of the cell and less abundant inside of the cell. So the natural direction of transport will be towards the inside of the cell. We'll come back to the thermodynamics of this in 1 second. In active transport, you're transporting the solutes against their gradient, so towards the compartment that is the most concentrated. And this is not favorable thermodynamically, so this means that this process has to be coupled to a process that is favorable. And so in that case, it's the hydrolysis of ATP into ADP plus PI, but it could be other types of so-called exogenic processes. And we'll come back to this in a minute. Another distinction that you may make, that other than passive and active, is whether the transport is mediated or not mediated. And mediated means that there is a transporter protein that is involved in the process, and that this transporter has to undergo some conformational change to allow for the transport, whether it's passive or active. Non-mediated means that either the solute can go through the membrane directly, or it can go through a channel that is made by a protein, but the protein doesn't need to change conformation, so it's basically just an open hole or pore through which solutes can go. So this kind of transport is possible for small molecules like gases, because the barrier that they will encounter to go across the membrane is not too large. But when it comes to polar or even charged molecules, usually you need a protein to go through. So this is the thermodynamic basis of what I just described. In these passive types of transport, the process through which the solutes are being transported is diffusion. And like we said, the solutes will tend to be transported along their electrochemical gradient. So this is the chemical part of the process that is due to diffusion. If you take the free energy of transporting a solute into the cell, then it's equal to RT log of the ratio of the concentrations inside and outside of the cell. And so here, if your concentration inside of the cell is higher than outside of the cell, this ratio is more than 1. so delta G is positive. So the free energy of transporting into the cell is positive, it's unfavorable, so then you're not transporting inside of the cell. So if you do the reverse thing, you'll see that transport into the cell is favorable when the concentration of the solute inside the cell is less favorable. And the driving force for diffusion is entropy, meaning that if the concentration inside of the cell is lower, there are more, if the concentration inside of the cell becomes higher, there will be more ways to arrange the solute into the solution, and so this is favorable entropically. Now, if the solute is charged, then the movement is not guided only by the chemical gradient, but by the so-called electrochemical gradient. So here you keep this chemical potential part, but you add this term, and then the delta G is the sum of the previous formula plus ZF delta EM, where delta EM is the electromotive driving force, so basically the potential difference between the two sides of the cell. So this governs overall the electrochemical gradient governs overall what is the free energy that will be favorable. Is it favorable to export the solute, or is it favorable to import the solute? But the other part that contributes to solute movement is, of course, crossing the barrier. So these equations, they work in the absence of a membrane. If you have just a solute where the solution is more concentrated on one side than on the other, then this mixture will homogenize over time. But if you have a membrane in the middle, then this becomes contingent on the solutes crossing the membrane. And like this figure shows, the partition coefficient from water into octanol is correlated to the permeability in a tissue. So this means that the more the compound can be inside of the membrane, the more it will get absorbed in a tissue. This is related to the permeability of the membrane. So this is not a perfect correlation, but it works fairly well, and it demonstrates that this part of the energetics where you have to transport the solute is important, as well as the electrochemical driving force that guides whether this what is the end product of transport. So water, we said, is very important. This means that there are dedicated proteins that transport water, and this is called aquaporin, and it's the same protein that I used when I was showing you the arrangement of lipids around the protein and telling you that these lipids are tightly associated with the protein. It's made of tetramer, so 4 proteins. Each monomer is made roughly of 5 helices, even though this one is broken. And you can see that water arranges in a single file inside of the protein. And this allows water to move in and out, again, depending on its chemical gradient, meaning its passive transport. And I don't have time to go through this, or I didn't have time to go through this in the lecture. It's really quite interesting how this protein is able to transport water without transporting protons. Protons are really important because, as you know, the proton concentration is related to the pH, and the pH governs many phenomena that are happening at the membrane and inside of the cell. So this type of protein really should not transport protons, and protons tend to be transported when water is transported because they are distributed inside of the water and they can circulate through this so-called Grotthuss hopping. So the protein makes sure that protons cannot be transported through the arrangement of the water file. And if you're interested in this process, it's a quite interesting phenomenon to look up. Okay, so about passive transport, I'm going to talk quite a bit about ion diffusion. The reason for talking about it is because we are very interested in ion transport in general in our research groups, but also because it's a very important phenomenon for physiology. So this is a prototype bacterial channel called KCSA. And basically at this point I'm going to go through explain what the structure of this channel is like and how it supports its function, which is to conduct ions and only ions and even only one type of ion. So like I said, KCSA, this channel is called KCSA, and it's called KCSA because K stands for potassium selectivity. So this channel will transport only potassium ions. And if you recall, potassium is actually larger than sodium, and sodium is very abundant in biological media. So how is this channel able to transport only potassium and not sodium? That has been kind of a long-standing question because it's so crucial for physiology. And the answer kind of lies in the structure and the dynamics of this protein. So KCSA is made of 4 subunits. Here you can see 2. There is another one here in the front of the screen and another one in the back of the screen, so it's assembled as a tetramer altogether. And each monomer is made of 2 transmembrane helices called M1 and M2, and a reentrant loop with a short helix called here the pore helix. And then this structure makes up mainly 2 regions. One is located here. It's called the gate, and it will be able to open and close. Whereas this is called the selectivity filter, and this is the place where ions are discriminated for or against. So it's because of this region that potassium ions can go through while sodium ions cannot. And here I have a movie showing this phenomenon. So this is a molecular dynamics long simulation where, sorry, I'm going to back up for a second, where you could see a focus on this region. So the molecular dynamics simulations contains the entire membrane protein with water and with a membrane, but then the movie is zooming in onto this region to show you how selective ion diffusion can occur. And here are the chains of the 4 subunits that are depicted, the main chains. And they're not depicted by but the side chains are actually pointing away from this structure. And here instead what is depicted is the carbonyl groups that are facing the ion permeation pathway. So I'm going to play the movie, focus on the ions that are shown as large spheres and the water molecules that are shown in between. So the typical mechanism that you can see here is that you need an ion to come to the bottom to be able to push the other ions through. This is called a knock-on mechanism. And you can see that there are times in the simulation where this type of arrangement stays for quite a while an ion will come from the intracellular domain and push the single fab. And so this doesn't explain exactly why this type of channel is selective, but what you can notice is that the potassium ions tend to localize and stay for a long time in regions where they are coordinated by a total of 8 carbonyls. And as I said before, sodium ions are smaller, so they tend to not stay localized in these regions, but instead they tend to be localized between 4 of these carbonyls, and this makes a less favorable thermodynamic arrangement, and this is one of the proposed mechanisms for potassium selectivity over sodium. I should mention also that maybe this is one of the first times that you're seeing this sort of thing. So we tend to think of structure as guiding function, but this kind of highlights that dynamics is also important, as you can reveal from computational methods like molecular dynamics. And actually, dynamical properties are thought to be important also in the selectivity process. So if you change the chemical properties of the amino acids that make this, you might change the dynamics of the transport process, and then you might change selectivity. I mentioned earlier that ion channels gate in the KCSA protein. I highlighted that the gate was at the level of the M2 helix towards the intracellular domain, and you can see here that there's a hinge region where the channel goes from closed to open. And in general, now not KCSA but different type of ion channels are gated by different stimuli. So they can be gated by a change in the transmembrane voltage. They can be gated by the presence of the binding of ligand, and they can be gated by mechanical stimuli, and many other things. What does voltage gated means, or what does ligand gated means? It means that voltage or ligand or other types of signals change the thermodynamic balance between this closed and open state. So these types of proteins will be able to be closed or open, but then if they're voltage gated, changing the voltage makes it more prone to open or more prone to close. Whereas when it's a ligand gated channels, it's more prone to open and close when the ligand is bound or not bound. So here's an example of a voltage gated ion channel structure. It's a kind of more complex assembly than KCSA, but it builds on the same type of architecture. You may recognize this part of the channel that is small, with two transmembrane helices and a reentrant pore domain. And this is very homologous, very similar in sequence and structure to the KCSA channel. But there are also other domains in this channel. Specifically, this region is going to be called the voltage sensor domain, for a reason that I will come back to in a minute. And as I was telling you before, when I talked about protein structure, usually these complex proteins in eukaryotes, they're not only made of transmembrane regions, but you also have kind of large domains that may be intracellular or extracellular. So here, this T1 domain, for example, is thought to be important for the tetramerization, which means bringing these four subunits together. And it's a peripheral domain, so it's located not in the membrane, but in the intracellular part of at the intracellular level of the membrane. So this is the sequence of a voltage-gated ion channel. Like we said before, the pore domain is made of these two transmembrane segments and the reentrant pore loop. And there's another auxiliary domain called the voltage sensing domain. This domain is made of four helices, as you've seen on the previous slide. And the S4 helix is very interesting because it is really quite different from the typical type of sequence that I was talking about that is favorable in the transmembrane region. This S4 segment has a large number of positively charged residues, which are depicted in here in green. Lysine, arginine, lysine, arginine, 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 arginine. And as we have talked about before, this is not supposed to be favorable in the transmembrane region. But remember that if it comes inside of a bundle, then it's possible to isolate all these positive charges from the unfavorable hydrophobic environment of the membrane. And indeed, this is what happens. So the S1, S2, S3, and S4 assemble together in a bundle, and S1, S2, and S3 have negatively charged residues on them, so that the positively charged residues of S4 can stably sit in a transmembrane position. This is what the zoom in onto the transmembrane or on the voltage sensor looks like. The positively charged residues are in blue, and the negatively charged residues of S1, S2, S3 in green are shown in red. And why is this going to be voltage gated? Voltage sensitivity comes from the composition of S4, which has all of these positive charges. And so what happens is when the membrane polarization, the voltage changes, then these positive charges, they're able to sense changes in the voltage and move up and down across the membrane. So this tells us why this is voltage sensitive. It doesn't really tell us why the channel is voltage gated. Remember what I was saying, voltage gated means that the balance between open and closed is modified by the membrane potential. And the gate is located, as we said with KCSA, is located at the level of S6, so the last helix over there. This means that this domain and the gate are communicating over quite long distance. This phenomenon is called allostery. It means that the stimulus is occurring at a region that is different where we're interested in the gating. So here's a molecular dynamics movie of the phenomenon. See how S4 is moving down in a stepwise motion, and you can see that this whole domain is moving quite a lot with respect to the pore domain, actually. And indeed, this then causes this signal to be transmitted to the pore here and to close it when the membrane is hyperpolarized and S4 goes down, and to be open when the S4 helix goes up and the membrane is depolarized. I didn't have time to go through this, so this is just to highlight that channels are not only voltage-gated channels can not only be activated by voltage, but they also undergo a so-called inactivation mechanism by which they are blocked. But I don't have time to go through it. This takes me to ligand-gated channels. So whereas voltage-gated channels were located usually along the elongated portions of cells because they're involved in propagating electrical signals, ligand-gated channels are usually located in the so-called, at the level of the so-called synapse in the membrane of the in the postsynaptic membrane, so in the membrane after the synapse. And the biological function for these ligand-gated channels is that they are able to bind chemicals like acetylcholine that are released into the synaptic cleft, and then when these channels bind this type of molecules, they are able to open. These channels are made of a quite different assembly than what I just said with voltage-gated ion channels. There are pentamers instead of tetramers, and each monomer is made of 4 transmembrane helices and a quite large extracellular domain. And the chemical that binds to open the channel is known to bind in the extracellular domain. So again, which will then cause the channel to open here in the transmembrane region. So this is another example of allostery, where the region where you are binding the ligand is quite far from the region where you are opening the channel. And so this means that information has to be transferred from one place to the other. And this is an overall important phenomenon that happens in many cases in biology, so to take note of. This is it for what I wanted to say about ion transport. And now I want to talk about this transporter-mediated transport. So remember in this plot we had passive and active transport. We're still in the context of passive transport, but I mentioned that for some solutes you need to have a change in the conformation of the protein that will allow for transport to happen. So this is meant to show a transporter in green, and this transporter is able to adopt different conformations, one facing the outside of the cell and one facing the inside. So if glucose is present outside of the cell, then glucose will be able to bind into the transporter when it's facing outwards. This binding will cause a conformational change, so this phenomenon will now be conformational change from outward facing to inward facing will be favorable, and this will allow dissociation of the sugar into the cell. When the transporter is open to the inside of the cell, spontaneously it will transfer to the outward facing conformation. This is called alternating access mechanism. As you can see, contrary to the previous case where we had a basic pore where ions could diffuse in and out, here instead we have a case where there's never a pore open. So either you're open to the outside or you're open to the inside, but you're never completely open. So this prevents then diffusion across the membrane. And this is important for rather large molecules. If you had a pore that would allow glucose to go through, then many other things could also go through. So you need to make sure that you have a tight binding and a cycle in which there is never an open structure to both sides at the same time. Here's going to be a movie to fix the ideas. So this is called GLUE3. It's a dimer. Each monomer is highlighted in a different color. And this is not molecular dynamics. This is a morph. And what a morph is the structure has been solved in the two states, outward facing and inward facing. And then we're interpolating between these two states. Glucose is out here, and you'll see it bind. Binding causes this conformational change in which this cleft closes and this cleft opens, and then glucose can bind in, can diffuse, unbind and diffuse, and this causes then the conformational change back to the outward facing configuration. Here's the same movie from seen from the inside. And you can really see that you can really see this alternating access mode where you never see an open pore. Always it's closed either at the extracellular level or at the intracellular level. Okay, so this is what I wanted to say about passive transport. Remember on this initial plot I showed you, there was also the case of active transport. And active transport is slightly more complicated because, as we said, the spontaneous process is passive diffusion. Active transport requires coupling to a process that is favorable thermodynamically because you're trying to transport solutes against their electrochemical gradient. So usually the proteins that mediate active transport are called pumps. And the source of energy can come from, or the energy can come from different sources. One famous one is hydrolysis of ATP. So ATP is a molecule that has very energetic bonds, such that when you hydrolyze these bonds, they give out a lot of energy that can then be used to transport solutes against their gradient. But there are other types of processes also that can give transport. One is, and we'll cover this one, is coupling to another process that gives energy, but it could also be transport of electrons or absorbance of light. So the really key player in this family is the NAK ATPase, sodium potassium pump. And I think it's interesting to know that we consume roughly 1/3 of the energy that we intake to make this pump work in our body. So it's really, really important. And the reason it's really important is that it separates potassiums and sodiums. And this is important, as I said before, in physiology, like in transporting ions in ion channels. So this is what creates this separation of ions, and then the ion channels will then transport ions along their gradient, right? So these two processes are coupled and important. So the transport cycle that the NAK ATPase goes through is quite similar to the sugar transport, in fact, in the sense that there will be an inward-facing configuration and an outward-facing configuration. But instead of binding sodium, this one binds sodium and potassium. So in the inward-facing configuration, which you can see here, sodium is able to come and bind, and actually 3 sodiums will come in and bind inside of the cell. Like I said, the transport of sodium outwards will be unfavorable because there's already a lot of sodium outside of the cell. And so you have to couple this to a process that gives energy. In that case, it's ATP hydrolysis to ADP. This means that the pump gets phosphorylated. And then when the pump is phosphorylated, it spontaneously can adopt outward-facing configuration. Sodiums are released, and potassiums can bind in. When the pump gets dephosphorylated, then you see you'll see a spontaneous transfer to the inward-facing conformation, where potassium is released to the inside. And one thing to notice is that you're transporting 3 sodiums outwards and 2 potassium inwards. So not only are you separating these ionic species, you're also creating transmembrane potential because you're exporting more charge than less. And this is the reason for creating a transmembrane potential, which, as you've seen, is important for setting the electrochemical gradient that governs the transport of charged species like sodium and potassium ions. Another type of process that can confer energy is absorbance of light. And I'm not going to go through this, but just know that there are proteins like bacteriorhodopsin that can absorb light, and when absorbing light, this gives energy and causes a conformational change that can allow protons to be transported. And then finally, another type of source of energy is coupling to a so-called exergonic process. So like I said earlier when I was, no, I didn't say this actually. So in the gut, one of the roles of this is to absorb glucose from food. So glucose is already concentrated inside of the organ, but you want to absorb more of it. And to do this, then you need to bring enough energy. And so this type of active transport exploits the sodium gradient. And the way it works is that it will import 1 molecule of glucose for 2 molecules of sodium. I don't have a movie of this, but as I've shown you in the previous case, you were exporting 3 sodiums for importing 2 sodium. You can also have so-called symport, where it's like glucose is piggybacking onto sodium being transported. And transporting sodium inwards is favorable. Why? because the sodium-potassium pump created such a big gradient with sodium being outwards. So naturally you have a source of energy then there that you've stored thanks to the sodium-potassium pump, which by the way was hydrolyzing ATP, which was a costly process. But anyways, through this process then you can import glucose in a way that is favorable thermodynamically through these sodium-glucose co-transporters. Here's an example of another antiporter, which uses sodium gradient to export protons. I'm mostly just showing this to show you a bit the variety of topologies of membrane proteins and the fact that they all try to perform the same function but in a way that is quite different. This is a morph again. You can see that these blue domains move relative to a quite static white domain. Sorry. And protons will bind to these negatively charged residues when when in this position, and then we'll export protons, and sodiums are being transported through the same route but inwards. Yeah, so this concludes the part on transport. In the original lecture I gave, I didn't have time to cover signal transduction, so I'll just say one word about it. And what I want to mention is just the G-protein coupled receptors, because they're so crucial and because Eric will talk about them in the lecture on drug design. And the way GPCRs work is that they're able to bind, like I said on one of the first slides, they're able to bind a chemical that comes from the outside and then cause a cascade of events downstream. So at this stage you don't need to know much more than this, other than these GPCRs are made of 7 transmembrane helices usually, and the key difference between transport and signal transduction is that here you're not actively transporting anything into the cell, rather you're using signals from the outside to cause a change inside. And this is a quite different way to conceive of the role of the membrane. But both of these types of processes are exploited by biological cells and are both important. And GPCRs actually are involved in many, many physiological phenomena in disease when they're mutated. And the most important thing to know is that the pharma industry is very interested in them because they constitute 30% of all drug targets. And so that's why there's so much interest, and well, why you'll cover this in a later lecture on drug design. This was all on GPCRs. I don't have time to go through it. And there are different types of signal transducers, and in that book they think you can also think of ligand-gated channels as signal transducers because they're binding, like we said, ligands from the extracellular domain. So this concludes the lecture. Key points are to remember how the membrane is made of a bilayer, mainly of a bilayer made of lipids. Membrane proteins can insert in these bilayers, and they can then perform a different number of functions. Mostly here we talked about transport, which remember can be passive, where it's dominated by diffusion, or active, where it should be coupled to an active source of transport. And membrane proteins also are very important for signal transduction, as illuminated by the function of G-protein coupled receptors, GPCRs. And here you have your study questions. And thank you for today.